So usually when I'm doing a class, I would really like if my students also can actually interact while we go on with the session, because that is important. And from my side, so I know that you guys are awake and you guys can hear me properly. So can you all like hear me properly at this point? Can you all like at least type a yes in the chat box? Because if not, I would think there's a problem with the connection and y'all can't hear me properly.
Yeah, they can hear you, Tanya.
Okay, nice. So with Dr. Dulaj and his institute, I have been affiliated and been doing classes in biochemistry for the past 2 years. For students who are like waiting CAT exams and repeat exams and all targeted to the second MBBS. So therefore, I should like emphasize this is not our first rodeo. Actually, we have seen students. We know what you all are struggling with, and especially like Dr. Dulaj mentioned, the second MBBS is one of the difficult most exams that are held in Sri Lanka. So if you are a medical student in Sri Lanka and you are going through the second MBBS, actually you all should be remarked as the cream of the Sri Lankan brains, of course, if you all can get through it. So I have the handbook here, which you all get from the KTU. So the second MBBS examination. So you all know the composition, right? How this exam is organized. So on behalf of you guys, can someone like unmute and tell the components of the exam? Don't worry, this is a very friendly session. I'm not going to test your biochemistry knowledge. Here, the main motive is for you all to tell how you all should face this, because knowing how to face the exam is also important as much as you all study and try to put all the information, all the syllabus components into your brain. Because no matter how hard you study, you have to do it in a smart way. Because like, now you only have like 3 weeks. So the 3 weeks should be optimized. So, and also these 3 weeks are the max, like the peak of your anxieties. So this is the time where y'all will be like, oh, my note is not good enough. I have to like photocopy it from a different person. Okay, the other person has a better note. Okay, that means I don't know much. All the second guessing and the anxiety, self questioning. So trust me, even we will be having those self questioning dilemmas which we will be facing during this prep time. So the best thing to do is pacing yourself during this time, and make sure you always keep yourself focused and grounded. Why I'm saying grounded is you have to be present at this moment and not get carried away, because during like the last 3 or 4 weeks before the exam, you think, okay, I have to collect this note, I have to make a proper note on this topic. So now it is not the time for note uh short note makings or borrowing notes and photocopy, because those photocopy notes you will never get the time to read or go through before the exam because it's just 3 weeks. But the key to get through the exam is in these 3 weeks, doing past papers over and over again. You have to do the past papers. One thing is the questions that you will get will be somewhat similar because for example, in carbohydrate metabolism, it's gluconeogenesis, glycolysis, and the glycogen pathways and all that. So somehow or the other, there's no new pathways that are going to come out of nowhere. It has to be one of those. And about uncontrolled diabetes or the fed state, fasted state. So these questions through the year, it will rotate. I'm not saying like it is going to repeat and this is how it is repeating. No. When you practice, you're actually revising the syllabus because one paper has 6 essay questions. So when you're writing those essay questions, when there are points that you don't know, or if you are not sure, you can just refer your notes. But you have to write. You have to compile your own answer for each and every question. And when you see the past papers also, I recommend do at least like 5 years backwards. When you do, you kind of see a pattern also. Okay, 2024, this particular pathway was asked in carbohydrate. And in 2023, it was this pathway. Okay, then out of this, there's a chance either this or this might come. So this analysis you can do only if you do the past papers. I hope you all get the point what I'm trying to point out. Because by doing the past papers, in an indirect way, you are covering the syllabus and you're covering the syllabus multiple times. When you do, say, you can finish 10 past papers. With those 10 past papers, you get to revise the syllabus 10 times. And you're always in the exam mode. If you're always sitting with a stack of notes and going through the notes without doing anything, it is not going to make any difference. Because it's like doing a sport. You have to practice. You have to practice, keep on practicing. Then only you will, on the game day also, you will actually play well. You can't be always strategizing and learning about it all the time. You have to do it. So it's the same thing with the MCQs and the SCQs, and especially the Viva. Viva, you have to open your mouth. Like, open your mouth in the sense, like, when you're studying, I hope you guys have a partner who's studying with you. So you all always should question each other. And another method I would suggest is usually when you're doing an MCQ, turn it into a Viva question. Usually, the students that we help usually know this. So when we are discussing an MCQ, for example, the MCQ is regarding the glycolysis. You don't just find the answers for that true-false question. You study around the question. So if you have a study buddy or a group of students who are like discussing, the discussion should be converted into a Viva discussion. So one can ask, okay, define glycolysis. Then what are the rate limiting steps in glycolysis? How many ATP are produced during glycolysis? And a very popular question. The importance of glycolysis in a red blood cell. And the glycolysis and the not the red blood cell and the HbA1c, the relationship, the glycosylated hemoglobin. These are quite repeated popular questions. So I did my second MBBS like 12, no, 15 years back. So, but I'm here teaching you guys. So why do I remember these particular questions is that when I do give these questions to y'all, I have seen these questions are keep on repeating, and these are very popular concepts which are being questioned from you guys. And like the thing in biochemistry syllabus is actually once you finish the second MBBS, you will realize, okay, this biochemistry syllabus is the shortest among the second MBBS subjects, because when you compare it with anatomy, physiology, biochemistry has like the shortest content. Biochemistry is difficult because of all the boring metabolic pathways. But once you learn how to crack it, it is not as difficult. How you crack it is you take the question papers, you see what are the concepts that this biochemistry department keep on testing you guys. And you prepare yourself according to those concepts. Because there's no other way around. In every exam, it's like that. So, according to the handbook, y'all will get 40 MCQs. And there will be 6 SCQ questions and 20 OSCE stations. And there will be a 7-minute viva. So altogether, the marks will come up to 90. And with your CAT exam performance, it will add another 10 marks. So the total will be out of 100. So I hope by now, all of y'all have an idea about how the second MBBS exam is going to be with the components. And of course, with the timetable is already published, isn't it? So y'all should be having a gala time of anxiety. But we all went through that. So what I'm saying is it is possible to get through the whole anxious studying and all that. But once you're done with it, you're actually done. It's freedom. So ideally, this should have been like 3 months back. You actually putting yourself the focusing to the studying, and you keep on focusing on the subject material and making sure that you're getting the daily working hours done. So at least I would say you have to like study like 10 to 12 hours, because you have anatomy. Anatomy is like a huge subject when it comes to the syllabus proportions. Uh, so do you guys have any questions that you all need me to like address? You all can type the question. So today we are like mainly focusing on the stress management factor. So to spice things a bit, I'll share a few model MCQ questions. So I'll be sharing a MCQ question. 10 questions. So maybe take 10 minutes of the time and try to come up with the answers. So these are like taken from the previous past papers. So I'll drop the folder into the chat. Okay. Uh, can you guys just like, either raise a hand or say yes if you all have like gotten the Mogul MCQ paper. So it's not a paper per se, it's a collection of 10 MCQ questions. Uh, take 10 minutes and come up with the answers. And we will be discussing this. Okay?
The chat. Hello.
Dot, Okay, can you all see the biochemistry Mogi paper or the Mogi set of questions? Hello.
Yeah, we can see this. Students can see this screen.
Okay, nice. So I can give 5 more minutes and we will briefly discuss the answers. Okay. You all can hear me, right? So, like when you all are typing the answers, like go with the stem letter also, like as if A, B, something like that. Okay, I'm waiting for y'all. Can you all, like when you all are sending the answers, send it to the main chat, because so that I can see the answers. When you all send it personally, I can't like go through all the chat windows to check if the answers are correct or not. And one important thing when you're marking the true false questions, it's that uh, in the multiple true false, you all get negative marks. So it is actually the marks are given out of 5 per question. So with each stem, you can score up to 1 to 5. Then also among, say, if you mark all 5 questions, you might mark all the 5 stems, and out of these stems, 3 you are like 100% sure, and 2 of it is like 25%. And these 2 questions turn out to be wrong responses. So these 2 will have 2 negative marks. So what happens? So no point that you mark 3 questions, 3 answers here. This will cancel out 2 of your positive marks, and you will only get 1 out of 5. So you have to be super careful when you're marking. So my strategy is usually I will make sure the first round I will mark only the stems that I am 100% sure. I will see if I can like guarantee, okay, these stems are 100%. So say there are 5 stems. If you can mark up to 3 stems that you are sure that, okay, I'm 100% sure about the 3 stems, but the 2, one is around 75% and the other one is 25%. This is the surety. So among these 5 stems, I would suggest on the first go, you mark the 3 stems that you are 100% sure about and leave the other 2. Because in the paper, among the 40 MCQ questions, there will be questions that you are like 100% sure of all the stems, all 5 stems. So if you're 100% sure, go and mark all 5, because you are like sure that, okay, this 5 This is like a section that you are 100% sure of. For example, there could be a question about water-soluble vitamins. So water-soluble vitamins or fat-soluble vitamins. So those are easy things, no? I think from A-level days, you know what are the water-soluble vitamins and the lipid-soluble vitamins. So sometimes you will feel, okay, I know all 5 of the stems. So in questions like that, you can go ahead. And sometimes you might be like practicing with the past papers, and there could be repeating questions. In the repeating questions also, when you see the question, you would feel, okay, this is a question that I'm familiar with. I have seen. Just because you have seen the question, don't blindly go and mark the question. Please read the question. I have seen so many students do that. Even when I like ask them to like do the question, I see you all do that without, okay, this I'm familiar with. I can remember the steps. So you are going to mark how you remember. But in the exam, it could be the similar question, but with different wording. So you have to read it properly. Because honestly, the repeated questions, getting them wrong, it's like you're being given the jackpot and you're not patient enough to cash the jackpot. And because of your impatience, you're losing the fortune, something like that. So back to the MCQ. Answering. So, and there could be questions where you only know 2 steps or 1 step. Of those questions also, only mark the step that you know 100%, whether it is true or false. Because there are questions that you will know either 4 or 5 out of the 5. So what you have to do is you have to exceed the 50%. So all you have to make sure is you have to score more than 50. So after you mark all the stems that you're 100% sure, see if you have marked at least 100 stems. So it's 40. 40 into 5 is. I hope you all have worked out the stems. So at least marginally you have to have 100 responses to reach the pass mark. But just marking the pass mark of 100 stems will not guarantee you. So if you have marked 100% sure stems, 100 steps in the 40 questions of multiple true false, then see if you can find other 25 responses, which you can be like around 75% to 100% sure in this range. Minimum, you have to mark 120 responses for a paper that will have 200 total responses. Any questions about this part? So I see that few of y'all have like answered the first question. So I'm going to discuss this. So the electron transport chain. The other aspect is when we talk about MCQ questions, I told you we should discuss it in a way that will prepare us for the Viva as well. So if I am to practice for the Viva, I would like ask my study partner, okay, define electron transport chain. That would be the first one. So what is the electron transport chain? Or in the Viva, you would be asked, okay, give me another name for the electron transport chain. So electron transport chain is also known as the respiratory chain. So by definition, it is the sequence of enzymes that carry enzymes and carriers in the inner mitochondrial membrane that are responsible for the transport of reducing equivalents from the substrate to the molecule oxygen to form water with the generation of ATP. So this is the last step of TCA cycle, where the reducing equivalents or hydrogen ions or electrons such as NAD, FAD will be converted to NADH and FADH and be releasing the energy. So in the definition itself, you get the answer for the first question. This takes place in the inner mitochondrial membrane. So the first response, this is false. Because this takes place in the inner mitochondrial membrane. The first step says electron transport chain occurs in the mitochondrial intermembrane. No, it's not in the intermembrane, it's in the inner membrane. So it is false. So to talk about more about this respiratory chain, this occurs in the inner mitochondrial membrane. The components, so enzymes such as oxygenase, dehydrogenase, oxidase, these enzymes takes place in this electron transport chain, and also a series of coenzymes such as NAD, FAD, coenzyme A, coenzyme Q, and iron sulfur proteins and cytochrome. So this is a place where cytochromes function. So cytochromes, we study the cytochromes in, we studied hemoglobin-like molecules where there's oxygen affinity and heme components. So in this electron transport chain, there are three, like complex 1, complex 2, complex 3, and complex 4. And what is the importance of this electron transport chain is, it is a final common pathway of aerobic cells in energy generation, and the carriers in the respiratory chain are arranged so that the spontaneous flow of electrons to molecule oxygen is ensured. And different substrates may use this common pathway because they are oxidized by the enzymes that use NAD or FAD as electron as receptors. So any pathway that generates energy, the last step would be this phosphorylation step. So the next step is coenzyme Q is the mobile electron carrier. So you have to know what are the electron carriers here. And what is coenzyme Q? There's a coenzyme A, and how this transportation usually takes place. So this is true. Coenzyme Q, it is a quinone derivative with the long hydrophobic isophrenoid tail. So this coenzyme Q acts as the mobile carrier. So that step, we can take it as true. Another very popular stem here, this Roctagon blocks complex 3. So this is false. Roctagon blocks complex 1. And it's an oxidative phosphorylation. Yes, it is true. Total energy production is 12. So this is false. The total energy production usually is between 30 to 32 per glucose molecule. So you all have to know the ratio how many ATPs are produced with one molecule of NADH and one molecule of NADH2. Usually 3 ATPs are formed from NADH and FADH gives out 2. So when you're studying about this electron transport chain, you have to study around not just the 5 stems. So you have to know the definition. So go with the definition. Components, importance. I'm sorry about my spellings, guys. Importance and functions, both. And the fourth one, the action of each component. So under these 4 topics, you should know. And since you all were asked about Roctagon, it's best to know what kind of drugs that can inhibit each and every complex. So Roctagon, barbiturates, and barbiturates are usually these sedatives. So it blocks the complex 1. And also when it is about electron transporting, you have to know the functions of each and every complex. So let's move on to the next question. Regarding the cholesterol reverse transportation. Okay, I'll give 1 minute. Share your responses with me. So in this topic about the reverse transporters of cholesterol, like the main reverse transporter in cholesterol is HDL. So in this question, what they are testing is your knowledge about the effects of lipoproteins, and particularly in this question, they are questioning you about the HDL metabolism. So synthesis in the liver and intestine in a disordered form. This is true. It is synthesized, HDL is synthesized in the liver and in the intestine, and it has a discoid shape. Transport cholesterol from liver to the peripheral tissues. Now, which lipoprotein functions as that? Usually LDL, VLDL is the carrier. Yes, very good. VLDL is the carrier of liver synthesized triglycerides to the peripheral tissues. So VLDL enters the microcirculation, and in the microcirculation, the hormone sensitive lipoprotein lipase acts on the VLDL and it converts to IDL, and from the IDL, the triglycerides are released, and by converting it into LDL, and the LDL is subsequently taken up by the liver, and the triglycerides in the circulation itself, it is broken to free fatty acids and glycerol. The free fatty acid in the peripheral tissues is taken up by the adipose tissue, and within the adipose tissue, the free fatty acids which were taken up from the circulation is re-esterified into triglycerides. So this is the fate of VLDL. So when I'm telling you this, I'm not even looking at the note. Yes, like there are a few points I didn't tell the about ApoB100. So in the fate, it is like, I'm sure you all got the notes and all from your faculty also. In the textbook also, in Lippincox and Harper's, there, there are specific details about how this ApoB100 comes into play and what ApoC does, ApoE, how it releases with the triglycerides and convert the very low density lipoprotein into IDL. Yes, these are minimum points, but overall you have to have the picture. Okay, what happens in VLDL? Okay, this goes and this, in the microcirculation, it is converted to this, and it removes the triglyceride portion, and it is converted to very low density to low density lipoproteins. So you have, that is what you say when you are studying metabolism. You have to remember the pathway, like a story. So what does HDL do? The reverse transporter, high density lipoproteins transport triglycerides from the peripheral tissues back to the liver. LCAT is esterified to cholesterol esters. Can someone type what is LCAT stands for? Yes. Lecithin cholesterol acyltransferase. So what do you all think of this stem? True or false? Okay, many of you all have answered well on this. So most of you all have studied lipid metabolism. So yes, it does not esterify into the, so usually what LCAT does is it esterifies the cholesterol into cholesterol esters. So this is false. Usually LCAT esterifies the free cholesterol. LDL plays a main role. No, that is also false. HDL plays a main role. Transport cholesterol from the liver to the peripheral tissues. That is also false. So remember, in the exam, you won't get all 40 questions, and don't expect all for, like what I was trying to say was in the during the exam paper won't be made out of 40 difficult questions, because in exams also there's a standard. Like they can't fail all of you. The paper has to be in a standard where there should be questions that you guys can answer. So don't expect the worst scenario or the worst outcome of your exam paper. Go to the exam with a positive head. Okay, it is going to be doable. Don't think it is going to be easy. This is the second MBBS, of course, but go with the positive mindset. Okay, this is going to be doable. And also, even in the most difficult question, all 5 stems are not going to be equally difficult. There's, there's always going to be that one stem which will earn the most bright student a distinction. And also there will be 2 or 3 questions for the mid-range student to just mark all 3 and get to the pass mark. So don't expect the worst outcome out of your preparation. You have to be positive. You have to always think, okay, I have put this many hours, I have done this many work. So the outcome has to be this. Be confident like that. So since we already did the lipid metabolism question, I will jump to question number 4 because we won't have much time. Which of the following are true false in hyperuricemia? Okay. Can you all like quickly type the answers into the chat box, please? Children, we have limited time to like go through our session. So it would be nice if you all can send your feedback, the answers as soon as possible. Question number 4. Okay. So back to the discussion. So hyperuricemia, the question is focusing on the, the question is focusing on the nucleotide metabolism. So nucleotide metabolism, purines and pyrimidines are the principal nucleotides that are in the biochemistry syllabus, of course. And out of these, so these nucleotides, we call them nitrogenous bases, which helps in formation of the DNA and RNA. So there are two groups, the purines and the pyrimidines, and the purine examples are adenine, guanine, xanthine, and hypoxanthine. So the hyperuricemia comes with the purine metabolism. So the first term, causes due to defecting PRPP synthase defects. So when you read the question, of course, you have to like work around the metabolic pathway. So PRPP synthase, so this, where does this enzyme come? It is the enzyme that activates the ribose 5-phosphate. So ribose 5-phosphate conversion to PRPP, 5-phosphoribosyl 1-pyrophosphate is the enzyme. So the preparatory step is the first step, and the rate limiting step is PRPP to 5-phosphoribosyl alanine conversion. So here, causes due to defects in PRPP synthase. So it is true. So overactivity of this enzyme will lead to increased synthesis of purines. So purine catabolism leads to increased uric acid, leading to hyperuricemia. Due to decreased synthesis of purine, it is false because, of course, the overproduction of purines leads to hyperuricemia due to increased rate of urate formation and decreased urate excretion. So this is also true. Due to HGPRT deficiency. So this syndrome, what does it call? Now this is also a favorite topic. So anyway, you guys are not answering a hyperxanthine guanine phosphoribosyl transferase enzyme. So this enzyme is important in the purine salvage part. So what this hormone enzyme does is converts hyperxanthine to IMP in the purine salvage part. So here due to HGPRT deficiency, true. And due to decreased amidophosphoribosyl transferase, false. Because what is the catalyzing step of this enzyme? Amidophosphoribosyl transferase. So in a deficiency in amidophosphoribosyl transferase will lead to reduced purine synthesis. So the reduced purine synthesis will not lead to a hyperuricemia because there have to be an excess production of nucleotides. So this question is not difficult at all. So they are questioning you from the nucleotide metabolism, but it's more surface basic things. Obviously, when you are like studying about nucleotide metabolism or any metabolism, you have to know the definition of the pathway, as in what the metabolic pathway does, and the components of it, as in at least the substrate, the first, the ingredients and the outcome, if it is catabolic or anabolic in nature, and the rate limiting steps. Rate limiting steps. And the fourth one, the regulation, the short term and long term regulation. These are the most important aspects that you have to study. So take any metabolic pathway. Under these four topics, you have to make sure you read, because in essay questions, in metabolism related essay questions, the first question is usually they ask you about a particular reaction. And the first uh, question is about naming the enzymes, something like that. So it is important, okay? So since we don't have much time left, uh, please complete the paper and like send your responses to Dr. Dulaj, and I'll get your responses from him. And in the meanwhile, tomorrow or day after, I will send you the answer script for these questions. So so I have shared the essay question. It's a Mogil essay question. I hope you all have received it, the file biochemistry Mogil essay question. So let me share it with you all. So usually this is how you will be asked. So there will be one question where the total aggregated mark will be 100. So each question will be out of 100. So there will be there will be 6 essay questions. 6 essay questions and 3 hours. You have to make sure you finish these questions out within the given time. That is one challenge. Because sometimes when there's a question that you know and you have studied very well, you invest so much of time in perfecting that answer, and at the end you might not have enough time to finish the rest of the essay questions. So this you have to be really careful. You have to make sure that you divide your time among all of the questions, because your paper is you have to answer all questions, not like there's no choice there. Like in the recent past, the questions, the paper, the essay question paper was of 6 questions, which you have to mandatory answer all 6. So the time management, you have 3 hours. You have to divide this among the 6 questions. So you can only take around 30 minutes per question. So ideally, what I would suggest is make sure in 20 minutes you finish the question and try to save the 10 minutes. Because I think you will be given 6 booklets, right? One question per booklet. So you will always have time to like come back and make sure you finish each and every question in 20 minutes. And at the end, roughly you will finish before 1 hour. So in 2 hours, you have touched all 6 questions. So with the remaining time, what you can do is you can revisit the questions that you feel like, okay, I haven't written enough. So you can perfect the question coming back to it. But this won't be the case all the time. Dr. Dulaj will give you more tips about the essay question writing. So in the biochemistry paper, when they question you about metabolism, mostly the first question will be about the biochemical reactions. They will allocate 10 to 15 marks on a question which you have to write so for example, they ask about glycolysis, and they ask, okay, what are the main regulatory enzymes in glycolysis? Or in nucleotide synthesis, in purine synthesis, they will ask, okay, what are the main rate limiting and regulatory steps? So you have to write the two ERPP synthase and aminotransferase steps there. Likewise. So the first part of a question will be like that. And then only you will be asked about a regulatory mechanism or the metabolism, the metabolic state in a fed state or in a fasted state, or a bit of clinical biochemistry with. So you can see the first question here. So this first question here is from so this question has ethanol metabolism and clinical biochemistry bit, chemistry on bilirubin metabolism also. And here, cystinuria, amino acid metabolism. Can you all type the answer for the first question here? Briefly explain 3 pathways. Briefly explain the 3 pathways in the human liver for the metabolism of ethanol. I don't need long explanatory answers, just the 3 metabolic, the 3 pathways. So yes, very nice. So in alcohol metabolism or in ethanol metabolism, it takes place in the liver because liver is the organ of detoxification. So the drugs, the substances, the toxins, everything is detoxified in the liver. So, so as alcohol, there are three main pathways. The first pathway is alcohol dehydrogenase pathway, which occurs in the cytosol of hepatocytes. So what happens is ethanol converted to acetaldehyde via alcohol dehydrogenase and converting NAD to NADH. So this is a major pathway when the ethanol intake is usually in a moderate consumption. So this leads to a production of large amount of NADH. So this alters the liver capacity to certain metabolic pathways, leading to hyperglycemia and triglyceride accumulation. So there have been different essay questions which they have asked about why this alcohol detoxification results in hyperglycemia, or how would the chronic alcoholic person uh, would end up with fatty liver disease. So it's the same concept they are testing. They are testing the alcohol detoxification pathways or alcohol metabolism. And in that scope, they are asking you all to relate in essay answer how this affects the gluconeogenesis in the liver, how it affects the fatty acid synthesis and the triglyceride accumulation or VLDL mobilization, how it is affected. So when that is why you have to do past papers to know what is expected from you. And the next system is microsomal ethanol oxidizing system, or we call it the P450 system. So it is located in the smooth endoplasmic reticulum in the liver. So what the reaction is ethanol and NADPH and oxygen to acetaldehyde. So this metabolic pathway takes place in people who are chronic alcoholic and who usually takes a very high amount of alcohol. Again, this what how this leads to fatty liver is because by trying to detoxify or with this ethanol detoxification metabolic cycles leads to a very high concentration of reactive oxygen species accumulation within the hepatocytes. So this ROS damages the hepatocytes with the increasing oxidative stress and lipid peroxidation, which accumulated effect leads to liver injury. So this is how you will relate fatty liver, and after some time these chronic alcoholics will face liver failure also. When if you get a question, usually they give a clinical scenario where the patient presents with jaundice, and for you to figure out which kind of jaundice this patient is having, and for you to figure out what has happened, as in, is it liver failure or is fatty liver disease? And based on that, you have to explain, okay, in which aspect in metabolic pathways is it this patient is suffering? Now, what has happened? So the ADH pathway is for It's the major pathway for mild to moderate consumption. When it comes to the chronic and high concentrations, it's the P450 system. And there's another system, the catalase pathway. So it usually occurs within the peroxisomes. And the catalase enzyme itself, it usually breaks down hydrogen peroxide. So here what happens is the ethanol and the hydrogen peroxide, which is accumulated in this in the above pathway in the P450, it converts the ethanol to acetaldehyde, and the hydrogen peroxide is converted to water. So this is sort of a minor pathway, but this contributes to the ethanol metabolism overall. So this is more actively seen in the brain rather than the liver. So this is the three pathways, and but this is a very easy question. They are asking you to briefly explain. So usually this is how the marks will be allocated. Usually you will get 5 marks for the introduction, and for briefly explaining each and every pathway, say around 10 marks each. And if you can write the clinical significance, like how the patient will eventually end up with fatty liver disease and liver failure, jaundice, all that, if you can add a bit of clinical significance, there will be another 5 marks. And that's how you can score the 40 marks here. So the next question, a 40-year-old patient with jaundice has increased levels of alkaline phosphatase and with the normal amount of AST and ALT, has dark urine and stools. Explain the biochemical basis following this patient. So here you have few clues here. For that you have to know clinical enzymology also. They are talking about alkaline phosphatase and AST and ALT. So can you all like type in the chat box where does this ALP is secreted from, and what is the significance of AST and ALT? So this bit is from the knowledge should come from clinical biochemistry. We don't have much time, so quickly. Tell me from where the ALT comes. Alkaline, ALP, alkaline phosphatase, and the AST ALT. So since no one else is answering, so alkaline phosphatase. So alkaline phosphatase is usually we can see it in a higher concentration when there's a problem in the biliary system. And AST-ALT is related to the liver. So here in the question, the first part is breaking down the question to figure out which kind of jaundice is this. So jaundice has three types: pre-hepatic, hepatic, and post-hepatic. Post-hepatic is the obstructive jaundice type. So here what you have to do is you have to differentiate the patient's condition between the hepatic jaundice and post-hepatic. I hope you understand why I'm not taking pre-hepatic jaundice, because there's no history regarding the patient's features of any hemolysis. So pre-hepatic jaundice is basically because of an increased hemolysis. So hepatic and pre-hepatic, post-hepatic. So obstructive jaundice is mostly after the conjugation on its way to the gallbladder. So it is usually a problem with the common bile, the biliary system or the gallbladder obstruction. So usually here in enzymology, you will see there's alkaline phosphatase increase, and it will be increasing conjugated bilirubin. Whereas if the liver is affected, so it's due to liver cell damage, which will lead to increase AST and ALT, which will serve as a biochemical marker here. So how will you explain the 30-mark question about how this patient is getting the jaundice in regard with the clinical information and the history which is provided to you? First of all, it's a 30-mark question. So you have to break it down. So you have to write a substantial amount to score all these 30 marks. First of all, you have to say what type of jaundice is this. And second point, you have to say why it is hepatic jaundice. It is because of the increased AST and ALT. And also, I would say you mention it why it is not obstructive jaundice, because it has normal levels of ALP. So here you show that, okay, I know about alkaline phosphatase also. So the essay question, you have to convince your lecturer, okay, I know about the subject material. That's about impressing your lecturers with your essay question, the answers. And the step three, and how this hepatic jaundice, the mechanism. So how do this hepatic jaundice occur? So basically there will be red cell breakdown in the reticular endothelial system, and this unconjugated bilirubin will be brought to the liver because of the ongoing liver damage. Because so in this case, of course, this person is so if we are relating it, assuming that he is an alcoholic person, so assuming only, okay. So this person might have had previous liver damage, but it is not given in the question. So without assuming, so with the AST and ALT levels high, it gives us a clue that there's some ongoing liver cell damage. So with the ongoing liver cell damage, the unconjugated bilirubin that is being brought to the liver cannot be conjugated by glucuronic acid at the optimum speed. So what happens here? The conjugating speed is affected. So it leads to jaundice. And the next question is about the color of the urine. So ideally, like in the above answer, you could have explained why it is hepatic jaundice because of the urine and justifying the urine color also. So you have to read the question and see if they are like trying to ask you about the next point, because no point of writing the same thing again. Yes. So because of the hepatocellular damage and the liver, the conjugated bilirubin will be leaked from the damaged hepatocytes. So the thing is, the conjugated bilirubin is water soluble. Unconjugated bilirubin is lipid soluble. So that's why unconjugated bilirubin can easily cross the blood-brain barrier. The conjugated bilirubin will be leaked to the circulation from the damaged cells, and it will come to the circulation where this water soluble conjugated bilirubin will appear in both stools and urine. So the conjugated bilirubin, when it is excreted in the urine, it will give a dark color, because in the bilirubin metabolism, the color of the stool comes from the urobilinogen. So usually when it is normally excreted from the intestine, the urobilinogen and the stercobilinogen, the stercobilin gives the color of the stool. That is why in obstructive jaundice, the lack of it give pale color urine. Sorry, pale color stools. In the because the conjugated bilirubin will be like sent back to the circulation. So there will be dark colored urine, but through the intestine and through stools, this stercobilin will not be excreted. So you have to always see the marks allocated to each and every essay question, and you have to answer accordingly. So since we are like coming to an end of this discussion, I can give you minutes for you all to post any questions that you all have. Do you guys have any questions regarding the exam and the biochemistry? Any questions? You all can type it in the chat. So any advice for Viva? So Viva is usually, you can always steer the Viva according to how you can answer. So when you're, say, if you're asked about glycolysis, like make sure not to say things that you are not too sure about. And that doesn't mean that you like, and it is not saying that you don't know. And the another thing is, do not lie in your Viva. Don't say false definitions or false answers. In that sense, it's okay to say that you don't know. But sometimes when a question is asked, at a glance, you feel like, okay, I don't know. But don't give in to that thought. Always take a minute to recall what is in your brain. Because sometimes you can, even if you don't know the Viva question at a at the given moment, if you like give it a thought for at least like 5 to 10 seconds, some kind of a clue will come out, because you all go to the exam with after studying and after so many sleepless nights of going through the subject material. So the content is there in your brain. Only thing is you need to give some time to recall it. So just after the question is blurted out of the lecturer, don't try to jump in and answer. Take 5 or 10 seconds to just breathe in before you compile the answer in your head. And if it is from a difficult section also, don't think, okay, I don't know, this is difficult. Because with your revision and the studying and everything, you at this moment, you should be able to like give some kind of answer. And the course of your answer, why the course of your Viva depends on the answers that you give. That is what I said in the beginning. You have the capacity to stir your Viva in the direction which you want it to go. So if you are talking about glycolysis, you give the definition. And then say if you are not ask another question, or sometimes the lecturer might say, okay, what is glycolysis? And then you go for the rate limiting steps. So when you when you ask the rate limiting steps, sometimes the examiner can go for the regulation side, or they can go to the metabolism pathway side, where they can ask about the substrates, the compartment, and the ATP that is produced, the number of ATP, like that. So when you're studying for Viva, don't study in paragraphs. Because in Viva, in that 7 to 10 minutes, you don't have time to give speeches. So it's like a very precise question where you give a precise answer. And don't be stuck with thinking about one question for a long time. The more questions that you attempt, you will get more marks. So sometimes in between, there could be questions that you can't answer. So it is okay to skip. You can say, okay, ma'am, I don't know. Or, sir, like, you don't know. But don't say you don't know for like questions before you even try it in your brain to think, okay, what, like, before you try to work it out, just because you feel the question is difficult, don't skip the question. And usually commonly asked areas. So I would say, what I usually say is, don't go to the exam without studying metabolism. And in the essay question, in the essay paper, don't skip the last question. It is usually the easiest question. And when you're answering the essay question, start with the question that you feel most confident about. So among the 6 questions, there's always going to be three or four questions that you are most confident about. And among that 4 also, there should be a question that is one of your favorites. You start from that, because the first question, like the first one, you write all what you know, like in the sense in regard to the question that is asked, and you compile it in a nice way. And the 6th question is usually from micronutrients, nutrition, and enzymes, the easy stuff. So that is why I'm saying, like, don't skip the 6th question. It's a very easy to score question. And usually the first 3 questions are from metabolism. So you will get a question from carbohydrate metabolism, lipid metabolism, and 3rd question probably the protein metabolism in combination with nucleotide metabolism. And in carbohydrate metabolism also, they love to ask about the metabolism regulation, insulin-glucagon ratios, and the biochemical basis of uncontrolled diabetes mellitus, or how the metabolic cycles take place in fed state and fasted state. And in protein metabolism, usually you could get a question where you are asked about ammonia metabolism and nucleotide metabolism together. So if you are being asked about ammonia cycle, the enzymes that are regulating the rate limiting steps of urea cycle, and then they might come into hyperuricemia or gout, the biochemical basis of gout, or you could be asked about these drugs that are used for the treatment of hyperuricemia and gout, this allopurinols, 6-metacaptolurine. So read more about this. And also in lipid metabolism, usually the favorite points are usually in fed state and fasted state, how this lipid metabolism take place. And don't go to the exam without reading fatty acid synthesis and detoxification, because those two are like the must know pathways. What else? Any more questions? And also read about the genetics where they will ask about PCR, RFLP. Like it will be a basic question around 30 to 40 marks. But make sure that you give attention for the metabolism. The carbohydrate metabolism, lipid metabolism, protein metabolism, and nucleotide metabolism. These are like the most important aspects. Like in fine sense, like don't go to the exam without studying metabolism. So anything else? You guys can type any questions that you all want to clarify. And also, Dr. Dulaj asked me to tell y'all that if you all want any assistance or you all have any questions, we are willing to answer through WhatsApp. So he'll soon be creating a WhatsApp group and you guys will be added to it. So yeah. So I'll wrap up the class. I hope this was beneficial for your exam preparation. Okay, let's call it a night then. Thank you so much for listening and paying attention. Good night, guys, and good luck for your exam. If we won't get to talk again, I hope you guys pass and get to continue the MBBS studies in the third year with the clinicals and all. You guys might think the second MBBS is the nightmare. No, the nightmare still goes on even after you become a doctor. But it's a nightmare that we willingly choose to live. So be positive about it. And trust me, you will never regret being a doctor because it's a very rewarding profession. So all the best. I hope you all will do well in the exam. And don't worry, guys, you all have 2 weeks. Revise as many as questions as you all can before you go to the exam and make sure to sleep well before the exam date. Okay.